Can you imagine a world where we can change our DNA as easily as editing words in an email? Though this technology isn't nearly as accessible, scientists are able to genetically modify our DNA more simply than you'd think. What may be even more surprising is that this practice dates back to the 1970s when researchers discovered how to clone DNA.
What that means is like taking one piece of DNA from one bacteria or one organism and sticking it next to another piece of DNA from another organism. And there was actually a lot of fear generated. This was about early 70s or mid 70s.
That's Dr. Raj Awatramani, professor of neurology and scientific director of the Transgenic and Targeted Mutagenesis Laboratory at Northwestern University. He says that the public's fear soon fizzled out when more information revealed that this cloning process wasn't nearly as dangerous as it seemed. In the 80s, one team of scientists evolved the method by injecting fertilized mouse eggs with DNA they had modified to include a growth hormone. The altered mice grew twice as fast and were 50% larger than normal mice.
That was the first idea. That was the first time it was demonstrated that you can introduce foreign pieces of DNA into a fertilized egg and have that animal grow up and carry that gene and express that gene and manifest some of the phenotypes driven by that gene.
This moment was revolutionary for genetic engineering. However, the main issue with the process is that the injected DNA couldn't be strategically placed within the embryos.
You just take your piece of DNA, stick it into an injector, stick it into a fertilized mouse egg, and it just sort of randomly integrates onto any chromosome.
Which isn't ideal for scientific research. Jump forward to 1989 when two scientists developed a new type of editing that allowed for more sophisticated manipulations to individual genes. And though this method was used for the next 20 years, Awatramani says that there were still drawbacks to the technology. The process took a long time, and only a few people knew how to use the complicated system. It wasn't until the early 2000s that experts began searching for a better way.
People had started to think, how can we make this more efficient? How can we make this process more efficient? And it was realized at some point, maybe even earlier than that, that if you introduce a cut into the DNA, into the region that you want to manipulate, that facilitates the manipulation of that entire region.
This process is called homology-directed repair. It's when cells automatically repair breaks in DNA strands. After its discovery, scientists believe that they could use this naturally occurring process to edit genes.
So then the question is, how do you deliver a cut to a specific region of DNA on some little tiny corner of some chromosome in the genome? How do you deliver that cut so specifically?
A question that is answered with the creation of zinc finger nucleases. These are groups of proteins that are engineered to cut DNA strands at specific locations. The issue is that cells don't always repair the DNA correctly. Sometimes they make mistakes, like adding extra base pairs to the strand. In order to make sure that the breaks heal how researchers want, scientists insert a template that directs the cells.
You make a cocktail with the template and the protein, and just stick it into a bunch of fertilized eggs, and at some frequency, that event will happen.
Sounds simple enough, but the first hurdle was actually getting our hands on the templates and the zinc finger nucleases.
2010, these zinc finger nucleases started getting sold by Sigma. I was super excited. I said, okay, I'm buying it immediately, because this is what we've been looking for, right? So I called them up. It's $25,000 for a zinc finger. I said, okay, you know what? I got to do what I got to do. I'm going to get it. Turned out it arrives in a tube about that small. It's like a tiny drop at the bottom of that tube, and it's $25,000. My student was trembling holding that tube because if that spilled or anything, that's it.
Despite the steep price point, Awatramani's experiment was a success. But how many researchers can afford to pay $25,000 each time they want to edit DNA? Finally, in 2012, CRISPR-Cas9 was developed, propelling the field of genetic engineering into the future.
And CRISPR is remarkably easy to use. And you don't need to buy this $25,000 thing from some company. You just design your own guide RNAs that are very cheap, very easy to make, very easy to obtain.
Though many people believe CRISPR to be a gene editing technology, it's actually a segment of DNA. The technology, called CRISPR-Cas9, uses a CRISPR DNA sequence to edit genes. Dr. George Church says that nowadays scientists can essentially change DNA however they choose. Church is a professor of genetics at Harvard Medical School, director of the U.S. Department of Energy Technology Center, and is regarded as the founding father of genomics. He says that gene editing today is widely used for engineering plants and animals.
Plants for agriculture use, animals for transplanting organs to people, and those are already making their way into preclinical and clinical trials. It's being used for curing sickle cell anemia, which was one of the first molecular diseases characterized many decades ago. And finally, we now have a potential cure. And the list goes on. It's certainly used a lot in research as well. And it works on essentially every organism.
Awatramani uses CRISPR technology in his lab to genetically modify mice for other scientists in order to speed up their research.
Let's say you want to know what the role of gene X is. So the investigator might come to us, we would delete gene X, and then we would grow mice that don't have gene X. And now if they are smaller, then you'll say gene X was involved in growth. Or if their heart is malformed, then you can say gene X was involved in heart development. So you can play these games by deleting genes, adding them back, or doing a variety of sophisticated manipulations.
And though CRISPR has become synonymous with the entire concept of gene editing, it's not the only technology scientists use to modify DNA.
We do massive scale editing without CRISPR using bacterial systems that do recombination, basically. And we can change the genetic code so much that we can make an organism resistant to all viruses. And we just published a paper on that this summer where we made the first organism intentionally designed to avoid all viruses. We did that by swapping two amino acids, serine and leucine. They're two of the most common amino acids, but they're very different from one another. When you swap them like that, the host is fine because it's in on the game, but all the viruses are completely messed up in every one of their proteins. It expects a serine, and instead it gets a leucine. And so those two amino acids are so different, it's really, really broken.
Church says that as far as they know, this organism is resistant to any and all viruses, even the ones we haven't discovered yet. In fact, its offspring will also be resistant. Now Church is moving this research into human stem cells. And while we may evolve into being virus resistant eventually, why wait if we don't have to? Gene editing allows for scientists to speed up evolution and specifically target certain changes. Church says this prevents any risk of running into accidents that often accompany the evolutionary process.
Evolution is random. And if you crank up the evolutionary rate of a random process, then you end up with death, basically. You don't end up with something better, you end up with something dead. Because there is you quickly get to a point where you've made a mistake. And if you make enough of those mistakes, then everything in the population dies. But if you use things like machine learning or rules, we can tell where we can make the changes that won't be wasted. That is to say, they won't go to neutral sites that have no impact, and they won't be deadly by avoiding things that are essential. You hit the things in the middle, which are likely to be impactful, but not deadly. And so then you can really crank up the mutation rate.
Along with speeding up evolution, Church says that we have the ability to select and focus on singular genetic outcomes. This allows for scientists to ignore other changes that could have happened during natural evolution.
But normal evolution takes, a million years to get a new species. We get things that are almost as significant, or maybe even more significant in a certain sense, in months rather than millions of years. For example, that one I described that's resistant to all viruses, it is essentially completely isolated from all DNA and RNA exchange with the whole rest of the world. So it's not only a new species, it's like a new kingdom. So that would have taken billions of years, not just millions of years. But this is just engineering. It's what humans have been doing ever since we were humans. We build wheels and fire and eventually jet planes. And these are, in certain ways, more amazing than building a species that doesn't exchange DNA, which at a certain level is a little wonky.
It may seem like the ability to edit our DNA has opened us up to both unprecedented risks and rewards. But Church says this isn't the first time society has felt this way. Back when diseases could eradicate entire populations of people, vaccines changed the course of humanity. Now, getting vaccinated is as easy as heading to your local pharmacy. However, Church does believe this technology should be handled properly and closely monitored.
one has to be cautious about all new technology and has to engage a broad sector of society that thinks about things differently. Because with new technologies, there are often winners and losers, even if it's a net positive, and you need to be very sensitive to that.
Join us next week as we dive into the ethical debate of genetically modifying plants, animals, and even humans. How can we regulate technology that can change the genetics of our future generations? You can find more information about Drs. Raj Awatramani and George Church on our website, radiohealthjournal.org. Our writer-producer is Kristen Farah. I'm Elizabeth Westfield. This segment is brought to you by Capital One. With one of the best savings rates in America, banking with Capital One is the easiest decision in the history of decisions. That's banking reimagined. What's in your wallet? Terms apply. See CapitalOne.com/bank for details. Capital One, N.A., member FDIC. Coming up next week on Radio Health Journal.
If you've changed the sperm and the eggs that make up that individual, you're not making that decision for one person, you're making that decision potentially for many.
A look into the ethical implications of gene editing.
Then how your grandparents' choices can affect your health.
It's a kind of biological memory that's etched on reproductive cells.
All that and more on Radio Health Journal.
I'm Nancy Benson, host of Radio Health Journal. If you enjoy listening to Radio Health Journal, you'll also like our sister show, Viewpoints, which covers a wide array of topics from education to history to the environment. Here's a preview of what they're covering this week on Viewpoints.
I remember seeing the giant sequoias, and they were so striking in their size, or that sunset, or the beauty of the face of my daughter when she was born.
Understanding the emotion behind the moments we remember in life. Then,
many people started realizing that there were also families and children experiencing homelessness.
As of 2020, there are close to 600,000 Americans experiencing homelessness. I'm Marty Peterson.
And I'm Gary Price. These stories in depth this week on your public affairs magazine, Viewpoints.
Listen to Viewpoints on your favorite radio station and subscribe and listen to shows anytime on Apple Podcasts and Google Play.
And that's Radio Health Journal for this week. Follow us on Twitter, Facebook, and Instagram to learn more. And check Apple Podcasts, Google Play, and Spotify for a library of past programs. Plus, you'll always find previous segments and information about our guests at radiohealthjournal.org. Join us again next week for another edition of Radio Health Journal.